When our body is infected by a pathogen, be it a virus or a bacteria, the pathogenic proteins or the antigens associated with these pathogens are recognized by antigen presenting cells. They engulf that pathogenic pathogens and display these antigens onto class 2 MHC, which could be further recognized by T cells. Now, also B cells with its antigenic receptor can also determine the epitope. Now, the T cell and the B cell would interact with each other, and T cell activates B cell. Now, also the B cell, after activation, would form an antibody secreting plasma cell. Now, you know that every day we are encountering several different pathogens, and against several different pathogens, it is required to form antibodies with different affinity, avidity, and different types. If this heterogeneity does not exist, then our body would get weakened and the pathogens would win the war, right? So, in order to do that, our body has several ways by which this antibody diversity is maintained. And in this video, we would look at several categories by which we categorized antibodies based on the differences between them in several locations. First type that we are going to talk about is isotypic variants. Now we know that antibody is a protein, so it would be definitely encoded by a gene, so that is immunoglobulin gene, and the immunoglobulin gene encodes for differently light chain and the heavy chain. And the heavy chain and the light chain genes have different variants, different gene segments, and with the permutation and combination, several different combinations of antibodies or different variants of antibodies could be produced. In isotypes, different heavy chain genes, heavy chain constant region genes are used to produce the isotypes. For example, the constant region is very different for different type of antibodies, for example IgA, IgE, IgD, etc. Now the C mu portion give rise to IgM, whereas C delta give rise to IgD, C gamma IgG, and respectively epsilon IgE and C alpha gives rise to IgA. So all these different uh, constant region gene segments would give rise to different isotypes of the antibody. Not only that kind of differentiate in terms of structure, but also their functions, because different isotype has different function. For example, IgG can pass through placenta and thereby provide immunity to the newborn. IgE is involved in allergic response, whereas IgM, IgG, they are involved in complementation pathway and thereby eliminating the pathogen. Whereas IgA is prevalently present in the secreted fluid like breast milk, saliva, tears, etc., and they also help to provide the immunity in the newborns. So depending upon the isotypes, the functionality, the diversity is huge. Another way of classifying the antibodies is by allotypes. Now, the term allo is somehow related to alleles. And once I tell alleles are the different versions of a gene, just like an Android phone. So, the Android phone, the Android system, Android OS is kind of a gene. And different versions of the Android, like Jelly Bean, like Marshmallow, these things are actually the allelic variants. Similarly, our IgG gene segments have also allelic variants, which code for minute differences in terms of amino acid, but they produce different antibodies. Now, definitely, the heavy chain and the light chain gene segments are having multiple alleles, and these alleles could generate minute differences in both constant region, mainly in the constant region of light chain and heavy chain. Now, let's say we have two antibodies, both are IgG antibodies, but in a constant region they have a minute difference, and as a result their structure is differing slightly, and these are going to be allotypes of one another because they're encoded by different allelic variants. Now let's just look at the nomenclature of allotypes. Now nomenclatures are based on like the heavy chain or the light chain name. For example, IgG IgG antibodies are encoded by gamma, right? The gamma constant region. So it would be named as gamma 1M and its variant number like 321, something like that. And for example, the light chain would be uh, uttered as either kappa, K for kappa, and then M variant 1 or 2, something like that. Now, so IgG-M1 and IgG-M3, these are allotype of one another. So if we look at this sequence, so these variations could occur at different region, either at um, the constant region heavy chain, constant region heavy chain has also multiple different domains, and also it can also present in the hinge region between the constant regions, and the difference lies in few of these amino acids. Now, last we talk about idiotypes. Now, idiotypes are different antibodies which are produced by different antigens. For example, there are two antigens here, antigen A and antigen B. Against them, IgG is produced. But both of these IgGs are different, and they are different in terms of their variable region of the heavy chain and the light chain. These hypervariable loop sequences are different between two antibodies of same isotype because different different antigen need to be recognized by minutely different antibodies, and their affinity should be also very different. So that is why idiotypes are also important. So if you like my video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.